Welcome to a mini-series of Neurosalience, featuring interviews with some of this year's annual meeting's keynote speakers.
Welcome to the OHBM 2023 Keynote Series as part of the Communications Committee. My name is Joanna Bayer, and I'm here with Dr. Andreas Horn, who will deliver one of the keynotes at the upcoming annual meeting in July 2023. And I'm very glad to have a few minutes to chat with you, Dr. Horn. And thank you very much for agreeing to this interview. And welcome to you as well.
Thank you so much for the interview. It's very great that you do this, and thanks for taking the time and the interest. Thank you.
Great. So let's just start with the first question. How would you describe your research to a non-scientific audience from the general public?
Yeah, great question. So I would say in a nutshell, what we do is we look at neuromodulation sites in the brain, and we often use invasive sites such as deep brain stimulation electrodes that are surgically implanted into the brain. And we look at which networks are activated in different patients. These are often patients with Parkinson's disease or other diseases, often movement disorders, sometimes OCD, depression, Alzheimer's disease, so various disorders. We look at the focal points in the anatomy that we modulate, and then look at which networks associate with that or connect to these stimulation sites. And then in a way, we want to find out which networks were modulated by the patients that did really, really well after surgery, in contrast to the ones where they didn't do so well. So in a way, we want to find out the optimal connections to modulate, for example, to treat Parkinson's disease. And we do so across various disorders and conditions.
Thank you. That's really super interesting. And what fascinates you most about your research, and what are the questions you would like to solve with your research, like on the long run?
I find it super interesting that we can use neuroimaging, often using the MRI, of course, and CT imaging to look at these things that will often have a very direct impact onto clinical practice. So I think that's quite unique in this field where, it's sometimes just subtle things we find. For example, we should place the electrode slightly more laterally or slightly more to the front of the brain structure we're modulating. But since, we will refine these targets, for example, but that might really have a direct impact on patients' lives, more or less sometimes with single studies, depending on the question and depending, of course, the sample size and so on. Usually has to be replicated and so on. But I find it so fascinating that in contrast to many other, neuroimaging domains, there is a more or less direct translational trajectory to clinical practice, which is unique, I think. Really cool.
Yeah, it's really applied. That's really great. You can see the direct impact, by the way. So it's very exciting to see that individual structural and functional connectivity profiles can be used to predict the outcome of deep brain stimulation in Parkinson's and beyond. And you've already said, it kind of alluded to that. How can those findings be used in a clinical context, and what can be done in cases where the prediction might not favor the patient?
Yeah, so great question. I think these findings, which networks are optimal to modulate for Parkinson's disease, for example, have a scientific value just to understand the disease better, right? That's not really clinically relevant, but then also to really find the optimal network and then target that network, right? So one trajectory is to identify the network based on deep brain stimulation sites, and then to non-invasively target the network from outside using, for example, TMS or tDCS. We've just done that in a study that's not published yet, but with tDCS, where it's a deep brain stimulation informed network that we then modulate with network-based transcranial direct stimulation, and showing an effect on clinical motor symptoms as well in a double-blinded design. So that in itself is already, a great translation where we can use the network information to modulate the same network elsewise. I think there are different examples also by Shan Siddiqui and Mike Fox, and depression doing the same thing, deep brain stimulation network, and then TMS. And I think also in itself, in deep brain stimulation, we can, of course, use this network information to find the same network in new patients before surgery by, for example, using DTI or fMRI to analyze it in a patient-specific manner, and then define the exact same spot where to put the electrode and which network to modulate. that is not yet being done because the barrier, it is invasive, neuromodulation and so on. Although maybe with some exceptions. So there are some, more anatomical networks such as the dentato-thalamic tract that can be targeted directly with deep brain stimulation. So I think for the future, it would be really the avenue where we can refine, a network targeting approach. And maybe one anecdote we could show, for example, in tremor, that hand tremor versus head tremor would maximally respond to modulating the hand region of M1 versus more axial regions of M1. So we could also use it to refine the exact body part and find the optimal stimulation site for that symptom going forward. But again, a lot is so far retrospective data, often validated by multiple cohorts, right? But what's lacking is really prospective clinical trials to apply this further.
So one target that has been historically and extensively used for DBS is the fornix, in particular in Alzheimer's disease. And one of your latest papers in Nature Communication actually investigates deeper into the mechanism underlying fornix DBS. So could you maybe summarize what the highlights of this paper were?
Yeah, thanks for the question. So I must say that big kudos on this main body of work goes to Andreas Lozano, who I think also held the TALIREC lecture last year at OHBM and showed some of the results already that were preliminary at the time. So he pioneered this approach to target the fornix for Alzheimer's disease. I can probably personally share that I was a bit doubtful, as probably he himself and others as well, how large are the effects and so on, before looking at the data myself. But then we could show a clear effect between where we modulated and then the ADAS-Cog improvement, so the improvement of the cognitive symptoms after 2 years of deep brain stimulation. So there was a clear dependency of where we modulated and then the outcome. That to me is maybe not proof, but a clear hint that there is something. Let's say the stimulation sites were not randomly distributed. It was not noise. It was really a clear thing. You modulate here, you get better. You modulate here, you didn't. And that was so robust that we could do, cross-validation, 7-fold cross-validations, 5-fold, 10-fold, and so on. It was always remained, significant. And we could show that in a local level and also a tract level. And I want to highlight this was amazing work by Ana Rios Infante, who's the first author of the study. And she was actually a master's student when she started this. So it's a really nice success story, I think, on that end as well.
Have functional or structural connectivity profiles shown better predictions for DBS outcomes? And why do you think that is the case?
Great question. So in fact, I can probably say that there is this ongoing informal challenge between Mike Fox and I of which is better. Mike Fox is a bit more favoring a functional MRI. I'm a bit more favoring structural MRI. But, we're, of course, great friends. And I think we both agree both are valuable. So it's more a historical thing. When we started together, I brought in the tracts and he's, of course, more an fMRI person. Mm-hmm. I think fMRI has a great advantage that it shows indirect connections as well. We have coverage across the whole brain always, right? So we always have a value at each pixel if we use resting state MRI, so that has practical advantages, statistical advantages. Tracts, I would say, are better resolved and a bit more direct measures of, we really know what a tract is, if it's a true tract, of course. But, we can look at the anatomy book. We know it's white matter connections. And so they might have very direct clinical value, while fMRI gives us maybe more a statistical global brain network estimate. So the hope would be that they have complementary information, and using both, we can explain more of the variants and build better models. And I think in the first study we published together, Mike and I, we actually showed that. So we could show that, the two things explained different sets of variants, and together we could explain more variants.
And then the next question would be, should functional or structural connectivity profiles be better derived from large data sets or from small ones that are precisely tailored to the target population? Which might be especially relevant, if you have specific populations like Alzheimer's disease or something, Parkinson's. Yeah.
Great question that hits to home. I think we get that asked very much because I maybe should say that we often use normative connectomes for our analysis. And the reason is often that the patients themselves don't didn't undergo DTI scanning or fMRI scanning. So we use the site of stimulation and often use a normative connectome to look at the connections. And of course, that's indirect. People could say, hey, this is not from the patients. This is not disease specific. Parkinsonian brains are different. All that is true, very much so. But it's still an ongoing question where I feel like, first of all, there's a lot to learn with normative connectomes still, right? So we can use larger cohorts using them. Then the signal to noise in these data sets are much better. So especially patient data of movement disorders, patients that move in the scanner are not, great. Yeah. So sometimes you get a lot of noise and buy into a lot of noise. And if you use patient-specific data, you can't scan patients for 18 hours, to get a great signal to noise. So you have to compromise. So that's that. And then you could even go further. I think there's a great neuron paper by the lab of Cameron McIntyre where they showed, if you manually draw the atlases of tracts by anatomist experts without even using DTI, only then can you really refine these very small tracts that we're often interested in, such as the ansa lenticularis, ossiculus lenticularis, and so on, from the pallidum to the STN, or the com fibers that orthogonally traverse the internal capsule. These are really hard to resolve with DTI, even in normative data sets. So my lab has come from maybe patient-specific, gradually realizing, we need better quality, we need more resolution, we need finer tracts, we need better definitions, and then have bought into more the atlas-based approach. But I'm not saying either approach is better. I think future will be a fusion of both. So we want to become patient-specific and meticulous in the way we define the tracts and networks. So it's still, there's a lot to do. And I think the answer is probably in the middle. Right now, a lot to learn with normative resources still.
So the brain is kind of a changing and aging organ. So what impacts do brain aging effects have on the mediating effects of functional and structural connectivity profiles to predict outcomes of DBS?
That's a great question. And I must say, we did not look so much into age, but we have a few studies that we publish that give indirect evidence here. So for example, we could show that if you just look at the definitions of coordinates of the electrodes in aging brains, age did play minor effect if it's just about defining ACP PC coordinates, let's say M and I space. And my gut feeling is that so especially in very elderly populations, age will play a role. But for example, what is much more important is in Alzheimer's disease, for example, the atrophy of the disease itself, right? Or sometimes also in Parkinson's disease. So I would say that disease specificity is probably more important than age itself. And we have, spent the last 10 years exactly looking at how to best register brains from different populations to each other. Of course, use nonlinear warping techniques, but also come up with manual refinement tools where we can really manually, as in Photoshop in a way, refine the fits between brains, because it is so important in our field. It's really about the millimeters. So we have a technique developed by Simon Oxenford, who is a bright PhD student in the lab, called Warp Drive, where we can really refine the overlaps between STNs or between these nuclei in the brain. And we do so very carefully, and we then focus on this very specific region in the brain, and that needs to fit. So I would say even in atrophied brains, and we could show that in the Alzheimer's paper, that without this technique, we would not see anything. We would not be able, with normal, registration techniques, to overlap the images well enough. But with these manual refinement techniques, we could. So yeah, it's not exactly the answer you were hoping for, I think, but it goes to that direction. I would say the disease specificity is probably more important than age.
Yeah, it's like the question is more born out of the fact that I've been doing like a lot of brain aging things. So that was yeah. So yeah, cool. And then the next question would be: the beneficial effects of DBS in Parkinson's disease and Alzheimer's are well established. But in the outline of your talk, you also mention other conditions such as depression or OCD, so obsessive compulsive disorder. What are the findings there, and which networks are involved?
Yeah, thank you. So I must indeed, I think the question you heard was more correct. So in Parkinson's, it is really established.
Okay. Yeah.
It's not. So it's very investigational there. So I wanted to emphasize that because it might be even one of the newest indications to do deep brain stimulation in Alzheimer's disease. And Parkinson's disease we've been doing successfully since the 90s. So very established, FDA approved, CE certified, and so on. So you mentioned obsessive compulsive disorder, where I can say there is actually also a humanitarian device exemption from the FDA, and there used to be one from the European Union as well until this year. It was not renewed for political reasons, not for actual scientific reasons. So there we can say it is on the way to establishment. So we find good effects in OCD and in many patients, not in all of them. We have published a paper in 2022 because you also looked at findings. So in 2020, where we could demonstrate that across targets, specific fiber bundle seems to be responsive to OCD effects, and that was true in different targets that we hit with electrodes. So one was the SDN region again, the other one was the anterior limb of the internal capsule, the nucleus accumbens. That finding has been replicated, I think, 5 times now by different groups. And it seems to be really robust. Again, a clear signal. If we hit this bundle, OCD gets better. We're now looking at specific subsymptoms. So, obsessions, compulsions, anxiety, depression, and so on. So to further understand which exact connection might mediate which symptom. But I think in itself, for at least the average OCD patient, that's one issue here, the heterogeneity of patients. We do see good effects. And then for depression, Helen Maybrück has pioneered the field, and then also Volker Koenen in Germany with a different target. And I would say, especially Helen Maybrück's target has now been not established, but there's good evidence, and there are bigger studies running. There's a study here in the US planned, and then also one in Europe, and on the other target, I think ongoing, where at least it's very promising that we might find an effective target for depression. So I would say, in some patients, the effects are amazing. And sometimes if the battery depletes, as Helen Maybrück would say, the patients rush into the OR and with, acute depression in a way, or mood effects to get it replaced, which is a good placebo control, right? You don't know that the battery depletes. And so I think in individual patients, absolutely clear effects, but then 2 of the bigger trials had failed. So it's a bit mixed, and we're currently, I think, in this phase of getting back to speed with depression, DBS.
Yeah. I've worked with depression, and it's super heterogeneous, right? Like the diagnosis alone is super heterogeneous. So it's really, really difficult.
I think patient selection is really important. So that's why, that makes it more complicated to roll this out once it's established to more centers. I think there are even people that would say that it works if Helen Maybrück selects the patient, right? Because she's so much experience, but transferring that to other centers is I think is tough. Yeah.
Assuming funding was not an issue, which direction would your research take in the next 5 years, and what areas would you and your team like to explore?
Yeah, great question. What's the dream, right, to do? I think I'm already at a center where we can do what I really want to do, which is Mike Fox's Center for Brain Circuits Therapeutics here at Harvard at Brigham and Women's Hospital, where the idea really is to translate into clinical practice, right? So that's probably the simple answer. What I would do would really to translate it and accelerate this translation, maybe leapfrog a bit, and go faster into patients, because that of course is costly, right? You need clinical trials and so on, and personnel to run the trials and everything. So, but I think there's so much promising effort there where we're really beginning to personalize deep brain stimulation based on the connectome, in a way that we think of disease, and not disease-specific networks, but symptom-specific networks. In Parkinson's, for example, we're about to publish one paper that segregates tremor versus bradykinesia, rigidity, and axial symptoms that, we can all hit with one electrode, but we could much more deliberately target each bundle based on a specific contact of the electrode. So I really think we can personalize it in patients that have a lot of this symptom. They would get slightly different treatment and so on. So that is clearly one thing to accelerate the pace of translation and bringing things into patients further. Yeah.
Yeah, that sounds great. Like, yeah, there's a lot of like effort to personalize things like, I don't know, precision medicine or like also fingerprinting. I think that would be great. Yeah. Do you have any activities that go beyond scientific work, such as science, communication, or related activities?
Yeah, thanks for asking that. So in fact, I do run a small hobby project in form of a podcast where I interview experts in the field of brain simulation. It's called Stimulating Brains. It's free. You can check it out. And it's been a great pleasure to be able to do that. For me, it's an excuse to interview experts in the field, and they then reserve time to talk to me, which maybe they wouldn't if I wouldn't have a podcast. So that's the main excuse to do that. And I've learned so much in that process. I think it has 30 episodes or something. And, famous people in the field of brain simulation have participated as guests. And I've learned a lot with that. And I hope to contribute a bit to also science communication and, broadening the impact of neuromodulation across the broader public as well.
I guess the community would be really interested because there's also the Neuroscience podcast, and
I love that, by the way. Big shout out to Peter Bendettini. So I listen to most episodes. I think it's a really great podcast.
Yeah, that's really great. Yeah, cool. Thanks for mentioning that. And then the last question is, what are you looking forward to in Montreal at OHBM in 2023?
OHBM is my conference, really. I really much enjoy connecting again with colleagues. It's also this year that a lot of people are coming. I still run a lab in Berlin and Boston, so 7 people in Berlin and I think a few also in Boston. And then we will have a few from Berlin coming over. We have a few from Boston coming over. We might even do a road trip back and forth together and potentially rent an Airbnb together. I have a baby son that probably will come with my wife, so that would be fun. Scientifically, I would say I'm connecting with colleagues. I'm very much looking forward to Anastasia Yendicki's tractography workshop. She held the keynote also last year and is a friend. And I think I read on Twitter that Davide Momi, who I really like, and Jill Meyer, both colleagues, Jill from Berlin and Davide, I think, is in Canada somewhere now. And on whole brain neurophysiology modeling. So I think these will be exciting workshops and symposia. But I haven't yet seen the full program. So I will be, of course, looking very much forward also to the excitement of giving a talk in such an audience. Never done that before in front of so many people, I think. So that will be very exciting and then just connecting with colleagues. Hopefully meeting you would be great to connect and these things. So will be fun, I'm sure.
Yes, I agree. Well, thank you so very much for this interview. It was super, super fascinating. And let's hope to meet at OHBM in July.
Thanks. Yeah. Great. Looking forward to that. Yeah, thanks.